In our previous video, we have seen what is a nucleic acid and what is the structure of a nucleic acid and what are all the different features and functions of a nucleic acids. In this particular video, we are going to see the two important types of nucleic acid which holds all of our genetic information, that is DNA and RNA. First, DNA. What is a DNA? DNA stands for deoxyribonucleic acid. Already we know nucleic acid are the combination of its monomer nucleotides. Already we have seen the difference between a DNA and RNA. The most important difference is in the sugar group, that is the pentose sugar. In DNA, the sugar group is deoxyribose sugar. DNA is the cellular molecule that contains the instructions for the performance of all cell functions. When a cell divides, its DNA is copied and passed from one cell generation to the next generation. DNA is organized into chromosomes and found within the nucleus of our cells. It contains the programmatic instructions for cellular activities. When organisms produce offspring, these instructions are passed down through the DNA. So this is the image of a DNA structure. You could see here very well there is a phosphate deoxyribose backbone. So a phosphate group joining with the sugar group of the another nucleotide and forms a phosphate deoxyribose backbone, or we commonly call it as phosphodiester bond. So this forms the backbone of one particular strand. Similarly, there is one more strand. These two strands are connected together by its nucleotide base pairs, that is adenine and thymine. So adenine, thymine, guanine, uracil, these four are the basic components which connects one strand of DNA to another strand of DNA, forming the double helix structure of a DNA. So DNA commonly exists as a double-stranded molecule, as I said before, with a twisted double helix shape. DNA is composed of a phosphate deoxyribose sugar backbone and four nitrogenous bases: adenine A, guanine G, cytosine C, and thymine T. In double-stranded DNA, adenine pairs with thymine and guanine pairs with cytosine. This is what we call it as complementary pairing. This complementary pairing is very important in formation of this particular structure. If this complementary pairing doesn't work in a proper way, it leads to mutation. For example, if adenine, instead of pairing with thymine, if it pairs with cytosine or guanine, it leads to a mutation at that particular site. So this particular image shows a clear double helix structure of a DNA. So you could see this is a single helix and this is the second helix. Both are joined together by these base pairs, which is nitrogenous base pairs. So adenine pairing with thymine, similarly a guanine pairing with cytosine of another strand. So this is how a DNA structure is formed. Now we will move to the second example, or the most important one of our nucleic acid, that is RNA. So RNA stands for ribonucleic acid. So the sugar group present here is a ribose sugar, which is having the oxygen group which is absent in our DNA, which is deoxyribose sugar. So RNA is essential for the synthesis of proteins, the most important one. The information contained within the genetic code is typically passed from DNA to RNA, then to resulting the proteins. There are several types of RNA. There are like 4 examples I have given here. The first one is messenger RNA. In short, we call it as mRNA, which is the RNA transcript or a RNA copy of the DNA message produced during the DNA transcription. Messenger RNA is translated to form the final proteins. So basically, what is this mRNA? So mRNA is the Xerox copy kind of thing of your DNA during the process called as DNA transcription. The second type of RNA is tRNA, that is transfer RNA. It has a 3-dimensional shape and is very necessary for the translation of the previous mRNA to produce the final protein synthesis. The next type of RNA is called as rRNA, which is ribosomal RNA. It is a component of ribosomes. Already we have studied about these ribosomes in Unit 1. Ribosome is an organelle present inside our cell, and this ribosome plays a crucial role in the protein synthesis. This ribosome is nothing other but a combination of rRNA along with few proteins. The last type is miRNAs, which is called as microRNAs. These are very small RNAs that help to regulate the gene expression. So basically these 4 types of RNA is very important for a production of a protein from the DNA. Now we will see the basic difference between a DNA and a RNA. So from this image you could clearly understand DNA is a double helix structure, whereas your RNA is a single helix structure. So it doesn't have another helix to join together. The second major difference is the presence of the sugar group. In DNA it's a deoxyribose sugar, whereas in RNA it's ribose sugar. The third difference is in the presence of its base pairs. For example, in DNA it has A adenine, T thymine, G guanine, C cytosine. Whereas in the case of RNA, the 3 base pairs are almost similar, that is A adenine, G guanine, C cytosine. But instead of thymine, it is replaced by uracil in the case of RNA. So this is a very important difference that is present between this DNA and RNA. There are multiple features that is differential, but these 3 are the most common difference between a DNA and a RNA. In the next video, we will see in detail about the different types of RNA and its role in the protein synthesis.